everybody checking their email and Moodle and all that sort of stuff. So you found there's homework. There's a homework assignment on there. Apparently I had the wrong date on it, so I updated it. It's not, what I have, like Tuesday, February 2nd. It's a week from today, 7th. Okay, so it'll be due in class, and we'll be doing a bunch of problems associated with that today. Hopefully we'll finish those so you can start working on those. So the way to look at this is, you can work together on these problems. One of the easiest ways to figure it out if you got it right is to do it independently with some other person. If you come up with the same answer, guess what? You probably got it right. Anyway, but you can work in groups, I don't care, but the key thing is turn each homework in with just your name on it, but also remember that these sorts of questions are likely to be similar to the work problems on the exam. So make sure you, yeah, no, 7th. So they're going to be due a week from today. So one is testing whether genes are located on separate chromosomes or are linked, which we're going to finish up today, and testing independent assortment. The other one is doing a Hardy-Weinberg equilibrium test, and then one is looking at how selection changes allele frequencies in one generation. I think we're going to get through both of those today, those last two, all three of those today, I think. So you'll get an idea of how to do that. Okay. And also just an update, the exam is 2 weeks from today also. I will be putting up on Moodle sometime soon old exams that are similar, so you get an idea of kind of the structure, multiple choice, work problems, short answers and essays, and an idea what they're like. And do you guys like study guides or not? Yes. Seriously? Yes. I never had study guides when I was growing up, so okay, I'll give you one. I always do. And then we'll do review sessions too. I'll try to set up some review sessions like the Friday or Monday before, and I'll record them in case you can't make it. Okay. So let's finish up today on, and what we're going to be doing a lot of is sort of, and did you do chi-squares last week in lab? Did you do those? Okay. So we're going to do hypothesis testing using various kinds of statistical techniques to test what we call null hypotheses, and we'll be doing that, and we'll be doing this first one today. So if you remember, we did this, you're going to be doing this in lab this week. You're going to be doing a bunch of Mendelian genetics. So remember the last time, what we did here is what's often called a test cross, and this was a cross between what we call a double heterozygote. In this particular case, it was big Y, small r, big R, small r, and that's a case of a yellow rounded parent times what we call a double homozygous recessive. And that's what a test cross always involves. One of the parents, whether it's male or female, it doesn't really matter. And that's homozygous at both genes or both loci. So that's a small y, small r, small r. And that was the cross. Okay. And you'll notice how I arranged these genes that influence these two traits, and I put them on separate chromosomes. And this is the idea, remember Mendelian second law, right? Independent assortment. The idea that alleles will assort independently of one another only if they're located on separate chromosomes. So what we're assuming here is, way I presented this, these two genes are on separate chromosomes. That is, they are not linked and they are not on the same chromosome. So what we can do is, and you'll be doing an example of this in corn, you can test this hypothesis and what your expectations are if these genes are located on separate chromosomes. And you can come up with this test cross, the basic idea. So let's kind of go through. I cleaned this up a little bit, and as I realized as I was going through this, I should have looked at it a little better. So go ahead and use this. So here on the left-hand column, we have the phenotypes that we observed when we did this, when we raised the offspring from this test cross, and oh, we got 1,000 individuals. Perfect. Real easy to do the divisions and everything. Real simple to do. Okay? And so here's what we observed of the 4 phenotypes, and what we have here also are the genotypes from this particular cross, for example, that you might get. We observed 499 of yellow round and 499 of purple wrinkled. And we only observed one individual of the two other phenotypes, the yellow wrinkled and the purple round. Does that look odd to you? Does that look like what you expect? And so what we'll talk about, the expectation under independent assortment that the genes are located on separate chromosomes, where did we get those expected numbers? What was the kind of expected ratio of those 4 phenotypes under 25% of each, right? So what we expected, if, like we showed in the previous graph, those genes are located on separate chromosomes, we expected 1/4 of each phenotype to result from that cross. Doesn't look like we got that, does it? So what you can do, the way to do that, of course, is you count up the number of individuals. This is the total number of individuals in your sample size, right? So there are 1,000 individuals. We come up with the expected numbers pretty simply, right? We expect 1/4 of each phenotype. So we take 1/4 or 0.25 times 1,000. And so our expectation, if these genes were located on separate chromosomes, and this is the cross we were doing, we would expect 250 of each phenotype. And then you guys know that right-hand column? That's how you do the chi-square test. And you're basically testing, given always the possibility of chance situations where you're going to get some slight deviations from expected, you're testing the hypothesis that the observed and expected are fairly equal, okay? And so that's how we do the chi-square test. And I'll probably give you that formula. You can memorize it. I'll give you that formula on the exam. I don't think it's worth memorizing, okay? And so you take observed minus expected, square each for each row, and here's the number you get, the chi-square, when you total all those things up. So the chi-square is summing all of these values, and it's 992. That's a real high number, isn't it? So if I were to, you did this last week, do you reject the hypothesis or fail to reject the hypothesis given this value with degrees of freedom? The number of phenotypes -1 give you 4. Yeah, this is an extremely strong rejection of the hypothesis that observed are similar to expected. And so something must be going on, right? Something's wrong here. What's wrong? Why are you getting such a deviation from the expected? Any hypotheses? Yeah, so the idea here was the hypothesis is derived from Mendel's second law of independent assortment that the genes are located on separate chromosomes. When you see a huge deviation like this, maybe that assumption is not true. And obviously, a lot of genes are located on the same chromosome, right? So what you might hypothesize here is that, in fact, these 2 genes, instead of being located on separate chromosomes, probably are located on the same chromosome. And anytime you see a deviation like this, that is usually when you do these test crosses, this is the thing. Okay. So there's something we can do even further, though. So when you look at those observed numbers, what do you notice? Is there kind of a class of, what strikes you about those numbers out of those 4 phenotypes? 2 are extremely common, right? And 2 of them are extremely rare. Okay? So that's the first thing you want to look at. Here are the common ones. And here are the rare ones. Now this is a little harder, and you have part of your homework is to try to derive this for a different cross. And I'm going to let you somewhat struggle through it and try to come up with the answer, and I'll try to show you how it's done here. But what this shows you, this is an example, and I'll go through it. These individuals in a test cross where you find this observed or not equal to expected, and you find 2 really common phenotypes and 2 really rare ones. The common phenotypes are almost always what we call non-recombinants. I'll just write that down, and I'll show you how we do that. So non-recombinants. And what do you think the rare ones are? Those are recombinants, okay? So let's go back. I'm going to do it on a separate slide. Let's go back to this original cross. Because what we've done here is we had this gene that influences seed color and this gene that influences seed shape being located on separate chromosomes, right? Now what we want to do is, well, maybe they're on the same chromosome, and I'll just show you how this might be arranged. Oops, I didn't want to do that. So how do we, whoops. Oh, I did have it on there. Oh, why did I erase that? Oh, well, it'll slow me down, right? Okay. So let's look at that male parent, the one, you know, that was the double heterozygote. There are a bunch of ways those genes could be located on the same chromosome together, and I'll just give you the answer. Here's the male. Now, instead of having two sets of chromosomes, we're just going to have one. There's the big Y, small y, right? And what do you want to put over here? It could be a big R up on the top, or it could be a small r on the top. I'll give you an A in the class if you get the right answer. Oh, okay, I can't give that many A's away. Okay. Okay. So let's look at this. Given this male parent, if this is how the two alleles from the separate traits are located on the same homologous chromosome, right? Big Y is associated with big R, and small y is associated with small r. What kind of gametes is that parent going to produce? How many kinds? Two in general, right? When that assorts, and that assorts through meiosis, right? Undergoes from diploidy to haploidy, right? You're going to get a big Y and a big R together in general. And what's the other kind? Small y and small r. What's the other parent over here? Yeah. Remember, the genes are still located on the same chromosome, and this is why we do test crosses. This is a small y, small r, small r. It only produces one kind of gamete, right? No matter what. And that's a small y, small r. Well, from that cross, what do you get? Two kinds of offspring. You get that. What's this bottom one? Small y, small y, small y, small y, small y, small r, small r. Okay. What are those two phenotypes? Yellow round, purple wrinkled. What are those? Top and bottom. So those individuals, those are the common ones from this cross. Those are yellow round. You got 499 of them, and you got 499 of the purple wrinkled. That's what you would get if the big Y and the big R are associated with each other on the same chromosome, and the small y and small r in the male parent are associated with each other on the same chromosome. How'd you get those other two? Yep. Okay. So what we've shown here is there's no recombination or no crossing over between those two chromosomes. So these are gametes that are produced when there's no crossing over or no recombination. Let me ask you, where would the crossing over have to be to get the other kinds of gametes? Where would it have to occur? Would it be occurring out here or out there? Or where does it have to be? It has to be in the middle. So let's imagine during doubling up and recombination and crossing over occurs, these chromosomes can be rejoined if you had something that occurred like that, right? Whoops, a little far. Okay. If those breaks occurred there and then they were rejoined, what kind of gametes are you going to produce? So these are what we call non-recombinants. What are the recombinant gametes going to be? Okay, one of them is going to be big Y, small r, right? And the other one's going to be small y, big R. Now, when you take those recombinant gametes, right, and cross those times this, what do you get? I'll let you do that. Guess what you get? You get these things, right? Okay? Okay. So your first homework problem is to do this. I think you also do this. I don't know if you do this in that level of detail in the lab this week, but so you'll get some chances to do this. So when you think about this, this is kind of what you have to think about. Do this sort of problem. So this is what we call linkage, and we're going to be coming back to that quite a bit. In fact, we violated, Mendel had some problems. He had some crosses where he did this and he was perplexed. He didn't understand the chromosomal basis of inheritance and all this sort of stuff, but he had some cases where he didn't get his expected ratios, and a lot of it was attributable to genes, in fact, being located on the same chromosome and not on, okay. And then there's a thought question at the end of your homework that I'm not going to address. I'm going to see what you guys come up with, okay, in terms of the frequency of recombinations. I'll let you think about that. Okay. The other great thing is I gave you a link to the Khan Academy videos. That is a great resource if you're a little fuzzy and a little under Mendelian genetics is a little puzzling to you. So, okay. Does that make sense? Kinda? Okay. So this will be reinforced a lot in lab. The homework will reinforce it, and so hopefully you'll have an understanding of it. And this whole idea of gene linkage is an important concept that we're going to come back to in the second part of the course too. And you'll be doing a bunch of Punnett squares in lab. I'm not going through that stuff, so you'll get familiar with the back, but refamiliarize yourself with Punnett squares. Okay. Now we're going to move a little bit away from sort of Mendelian genetics to a much higher level. And what we're going to be looking at is basically we're going to introduce you to population genetics. And what we're going to try to do is pretty simple. We're going to set up kind of a simple null model, which is basically what we expect when there's no change in the frequency of alleles or genes within a population and all the assumptions we have to get this, what we call an equilibrium. So we're going to be doing a lot of calculations today, and it can be a little overwhelming at times, and I understand all that. So what we're going to really try to do, a lot of the homework is to reinforce this. And again, I really don't like to memorize a lot of things, but a lot of these steps do require the path and understanding how you get from one step to the next. I'm the sort of person when I grade, if you kind of mess up, one part of it and you kind of get a calculation wrong and it cascades down into the rest of the part of the question, I don't take off a whole lot for that, right? So I think if you follow and know the steps, but you just made a slight calculation error, made a little boo-boo in the middle, early on, I'm not going to penalize you for that. So I try to minimize these sorts of things. But we're going to be spending a lot, what we're going to basically be doing this, I don't think we're going to get through this today. here's kind of the questions and what we're going to be trying to do this week. And we're basically, how do allele frequencies, so we had the example of the yellow, all those. So we're going to be looking at allele frequencies. We're often going to just refer to them as A1 or A2, fairly generic things. Is how do allele frequencies at a single gene or locus change over time in the absence of natural selection and other evolutionary processes? What we're going to be developing is a pretty simple null model that we call the Hardy-Weinberg stable equilibrium model. Was that introduced in biodiversity at all? Does that sound familiar? Kind of. Okay. So what we're going to introduce is what's called a null model. That is, the expectations, what our expectations are when there are no evolutionary forces acting on a population, on the genes in a population. This is what we call the null model. And there are a whole series of about 5 major assumptions that usually are violated or not true. And then we're going to start talking about how selection changes allele frequencies, how mutation changes allele frequencies, how gene flow changes allele frequencies, how non-random mating changes allele frequencies, and genetic drift, all these other things. So then we'll be moving on. What we're going to try to do today is how do different kinds of natural selection, what we call dominant directional, recessive directional, heterozygote advantage, and negative frequency dependence, affect allele frequencies. And we're really going to be focusing on is an equilibrium reached, and we'll be defining what an equilibrium is reached and how quickly is it reached. So as we go through, we're not going to get to heterozygote advantage or negative frequency dependence today, I'm pretty sure, because I'm going to be doing some problems. But as we go through each one of these, what I'm really going to try to emphasize is, how are allele frequencies affected? Do they reach an equilibrium, and what influences the rate of it? And what we often do is we're always often comparing these different models. So does dominant directional selection result in a faster change than what we call codominant selection? We'll be looking at examples of this. And this has a lot of applications in a lot of areas of biology. So I think it's kind of an important thing to talk about. Okay. We'll be looking at various kinds of traits in populations. Sometimes we have what are called discrete traits. So here's aloe, how it spirals, whether it's counterclockwise or clockwise. This is what we call a discrete trait. It's one kind of phenotype or another. It's very discrete. There's not this continuous variation in a population. We'll be focusing mostly in population genetics on sort of single locus discrete traits that only have, a couple possibilities in terms of phenotypes or genotypes. The other thing is we will later in first part of this course, we'll be looking at what we call quantitative traits. And so human skin color is a classic example of a continuous trait. That is, it ranges from, it often has a normal distribution. There aren't discrete categories. It's hard to categorize one thing versus another. It's a continuous trait, okay? And so we'll be looking, as we get to quantitative genetics, at what we call more continuous traits, and they require a little more complicated analysis. So what we're doing, this is kind of changing how we're thinking about things. And as you do your Mendelian genetics and you do your problems, that's what we call kind of individual level thinking. That is, what gametes and offspring are produced in what frequencies from a given pair of parents. So here's a classic thing. And we can come up with expectations about, given just a single gene with 2 alleles, what proportion of offspring of each genotype and phenotype you expect from this cross, say between 2 heterozygous parents, right? And this is kind of at the level of 2 individuals and their offspring. We're going to be now focusing on sort of population level thinking, so kind of at a higher level. And so what we're doing is, how do the characteristics of the population change over time as a result of evolutionary processes? And so here, what we're looking at was, we're not focusing so much on the individuals, but here we have a group of 6 genotypes in a population. So often we have thousands of individuals in a population. And what we're interested in knowing is, this is time 0, this is one time frame, 1 generation later. What is the change? Is there any change in allele frequency or the frequency of genotypes in a population as a result of evolutionary processes. And we're going to try to understand how these changes occur, what influences the rate at which they occur, and all these sorts of things. So we're going to be moving more towards a population level thinking. So what do we mean by an equilibrium? And we'll be spending a lot of time on this and looking at changes in allele frequencies from a starting time to an end time, and what's the outcome. So there are various ways to look at this. Here's a kind of a cone, and here we can define various kinds of what we call stable or unstable equilibria, okay? So for example, you can see here, this is what we might call, here's a ball in a cup like this, right? Is it going to be able to move anywhere? Well, unless you perturb it, okay? You could perturb it away from this position, and you could try to move, you could swirl around, but where's it eventually going to end up back any time later? Back at that bottom point, right? So this is just kind of a conceptual way to think about it, but stable, when we think about stable equilibria, what we mean is it does not move from this point. If you move it from that point, it always returns back to that original position. And this is the whole idea of Hardy-Weinberg. This is what Hardy-Weinberg is considered a stable equilibrium, that you can perturb a population away from a Hardy-Weinberg equilibrium, but it doesn't take very long, usually a generation, to return back to that equilibrium, and we'll be talking about that. Okay. And Hardy-Weinberg is kind of a classic example of a stable equilibrium. There are lots of different kinds of selection models that also result in stable equilibria too, right? That often result in particular alleles or genes being favored, that go to fixation in a population and do not change if we assume some other things. Unstable is, that's if you can stay right on that point, right, you're not going to move, right? If there's no perturbation or movement away from that top of this inverted cone, right? But what happens if it moves in any direction? Well, it'll move even further away from that equilibrium point. So that's often what we call an unstable equilibrium. It's kind of an equilibrium point, but it's extremely unstable. So anytime you perturb it away from that resting point right there and it goes in that direction, then it goes down and off and moves away in a very unstable way. So this is just to kind of conceptualize when we talk about equilibria. And we'll, as I go through each one of these models, I'm gonna try to outline, did we reach a stable equilibrium, right? Or is it unstable? And how long does it take? Okay. So let's start thinking about Hardy-Weinberg and how we think about the absence of changes in allele frequencies over time. So here's what we're imagining. When we model these changes in the frequency of alleles, and in this particular case, we only have 2 alleles in the population. What are they? A1 and A2, they're represented by different colors, okay? So this is a single gene or single locus with 2 alleles, and that's it. That's as simple as we can get, okay? So we have parents, right? We imagine that parents contribute gametes to what we call a single large gamete pool. Now, this might be examples of things that have like external fertilization, right? That spew their gametes out into the environment, especially in a lot of aquatic organisms, marine organisms. Not all things have this, obviously, but we can imagine parents then all undergo meiosis during a mating period or some sort of reproductive period. They spew their gametes out into this sort of mixture, and then those gametes can recombine, right, to produce offspring. So we imagine that parents contribute to gametes. This is how we're going to model Hardy-Weinberg. We imagine that parents contribute gametes to a single large gamete pool, and then pairs of gametes out of this gamete pool fuse, right, to produce new offspring, and it's done in a somewhat random way. And what we mean by that, it's not that A2 gametes are more likely to fuse with one another, right? Or A1 and A1 gametes are more likely to fuse with one another, that they basically fuse at random, and it's based on their frequency in the population. They'll fuse it in somewhat of a random way. The other possibility is another non-random fusion event would be if A1 and A2 were more likely to fuse. So what we're assuming is this sort of random fusion of gametes, and what do we get? And we're not going into all the details and the derivation of this model. It's a fairly simple model. But here are the three important conclusions from this Hardy-Weinberg. And we'll show you how we calculate and do all these important things. And we're all outlining the 5 major assumptions that are necessary for Hardy-Weinberg to work. But here are the 3 important conclusions from the Hardy-Weinberg theorem that was developed in the late 1800s. Allele frequencies do not change over time in the absence of evolutionary processes. Okay? So you can't have evolution. Here's the other key take-home message. Given allele frequencies in this random mating or this random fusion of gametes, we can predict the equilibrium genotype frequencies, and we'll be showing you how we do that. These are referred to as the Hardy-Weinberg equilibrium frequencies. This kind of gets to the stasis part, or the equilibrium part. If there are no evolutionary processes operating, a gene or a locus that is not in Hardy-Weinberg will reach Hardy-Weinberg equilibrium in 1 generation. You're actually going to be doing some experiments with beads and things to demonstrate this, not in lab this week, but next week. So you'll be doing a whole lab with beads and imagining this process, and you'll be demonstrating Hardy-Weinberg, and then you'll be actually imposing selection on these bead populations, and you'll see what happens. So you get some. This will be reinforced next week in lab. So those are some of the. I know probably doesn't mean a whole lot now, but these are the 3 important conclusions from Hardy-Weinberg. Here are the necessary conditions for Hardy-Weinberg to work in a population. We're assuming natural selection is not acting on that gene, and we'll be going through examples of that. Individuals mate at random with one another in the population. There is no preference for mating with individuals that have similar or different genotypes. So this is the random mating process. We're assuming no mutation is occurring, right? We're assuming that A1 is not mutating into A3, A1 is not mutating into A2. We're assuming there's no mutation occurring at this particular gene. There is no migration of individuals into or out of the population or a closed population. This is the idea that there's no movement of individuals or gene flow of individuals. Genes are not moving between populations. These are discrete closed populations. The last assumption is that the population size is effectively infinite, so that chance fluctuations in allele frequency don't occur. Whoops, that's don't occur. Take that else off. Okay? So there's no genetic drift. So what we're going to be doing is we're going to be showing you what happens to a population when each one of these assumptions is no longer true and how it changes the allele frequency. So we'll be looking at how natural selection occurs, how non-random mating occurs and affects gene frequencies, how mutation influences gene frequencies, how gene flow influences gene frequencies, and then how genetic drift influences gene frequencies. But we're setting up this basic null model in the absence of all these factors first. Okay, so I'm not going to go through the derivation of the equilibrium and all that sort of stuff. I don't think that's important. Let's just assume we have 2 alleles, A1 and A2, and we're going to refer to these alleles, A1 as P, basically, and A2 as Q. Given you have 2 alleles, there are only 3 possible genotypes, right? Assuming no mutations or no gene flow or anything like that. Here are the 3 possible genotypes: homozygous for A1, heterozygous, and homozygous for A2. So what we basically do, Hardy and Weinberg developed the model, and they said the observed frequencies of P and Q are determined from the genotype frequencies. So we're going to go through lots of examples of this, but what we're saying is that the frequency of the P allele or the A1 allele is basically the frequency of that genotype in the population plus half of the frequency of the heterozygote. And basically the way to think about it is this: you're just taking that by half because you're basically getting rid of the A2. So the way to figure out the frequency of the A1 allele is to take the frequency of the A1 genotype and what is half of the frequency of the heterozygote in the population. Okay? Q is defined as the frequency of the other homozygote, A2 in the population, plus half of the frequency of the heterozygote. And obviously, you can imagine each one of these have to sum up to some value, right? And they obviously have to sum up to 1. So one of the key things as you go through this process and you're thinking about exams, and you're trying to figure out how to do things really quickly, right? To give your time more time to do other things on an exam, always remember this concept, right? Once you solve for P, right, do you really need to go through that laborsome calculation of this? No, just take 1 minus P to give you the value of Q. Okay, so I'll show you how to do those sorts of things. Okay. From this, and there are lots of steps to get to this point, and you don't want to see this, and it's in your book if you ever want to do it. To get to this point, the Hardy-Weinberg equilibrium frequencies are defined on the basis of these allele frequencies in a population. And this is kind of the magical Hardy-Weinberg equilibrium. It says the Hardy-Weinberg equilibrium frequencies are pretty simple. The expected equilibrium frequency of A1 is that P squared. The expected equilibrium frequency of the heterozygote is 2 times P times Q, and the expected equilibrium frequency of the other homozygous genotype A2 is Q squared. Okay? So what do you really want to be able to do? You want to be able to, the first thing you have to be able to do to get these expectations right is to determine what is the value of P and Q in the population, given sort of the frequencies of genotypes in the population. So keep that in mind. Okay. So now I'm going to go to the Hardy-Weinberg work problem, and this PDF was on Moodle. You're going to be doing, your second homework assignment is this exact thing, okay? It's going to be a different numbers and different outcomes, but what I'm going to show you is how we do this. So let's start at this. Here's how we do these Hardy-Weinberg calculations. And this is actually a gene that's kind of, it is a human gene. I'm not going to go into all the details right now. But here again, we have, here's one of the homozygotes. Here's one of the alleles. One of the alleles is PAV. The other allele is AVI. So here's the PAV homozygote. Here's the heterozygote. Here's the AVI homozygote. Okay. Again, I'm going to make it simple for calculation purposes. Use 1,000 individuals. You probably have to use a calculator. I may not give you just 1,000 individuals, but just to show this. So what we're showing here is, here are the number of individuals that were observed in a population at this single gene with 2 alleles. So we have 1,000 individuals, 400 were of this genotype, 200 were heterozygous, and 400 were the alternate homozygote. We refer to these often as big P, H, and Q. These are the sort of observed number of individuals. And from these, we can determine what's called the genotype frequency. So here are the 3 genotypes. How do we get to that point? Yep. So the basic, you determine the frequency of each genotype by taking the number of individuals, right, in the population divided by the total number of individuals in the population. So 400 divided by 1,000 says the genotype frequency of PAV is 0.4, or 40% of the individuals in the population are PAV homozygotes. 200 divided by 1,000 gives you 0.2, or 20%, or the heterozygote, and this is, and obviously, if you get these 2, guess what you can do? That sums up to 0.6, right? So do you need to do that calculation? No. You know, it all has to sum up to 1, so that's 0.4. Okay. So here's the next, just kind of have to memorize this. Given these genotype frequencies, we can determine the allele frequencies from that previous formula. So we're going to say PAV is equal to the small p. Okay, we'll just say that by convention. And Q is the allele frequency of the frequency of the AVI allele. Here's how we do it. You basically take the frequency of that genotype one of the homozygotes, which we call P, plus half of the heterozygote. So the frequency of the P allele or the PAV in the population is P, 0.4, plus half of 0.2, right, gives you 0.5. So in this calculation, the frequency of the PAV allele in the population is at 0.5 or 50%. You can do the same thing with the Q or the AVI, right? And you can do the same exact calculation. You're going to get 0.5. Again, if you want to do it quickly, just take 1 minus 0.5 and you'll get the frequency of the other alternate allele because they all have to sum up to 1. Okay, and that's kind of the key thing to remember is P plus Q always equals 1. Okay, so now what do we do? Now we have, these are kind of the observed allele frequencies in the population, right? 0.5 and 0.5. We can then take that and you do this, and these are the Hardy-Weinberg expected genotype frequencies. So this is P. Just plug in those values in the P squared, 2PQ, and Q squared. So this would be, what would the P squared be? 0.5 times 0.5 or 0.25, right? 2 times we'll do this next in the next slide, but 2 times 0.5 times 0.5 is 0.5, and Q squared is 0.25. Okay, so it's as simple as that. Just kind of got to remember that equilibrium frequency based on the allele frequency, the equilibrium genotype frequencies based on allele frequencies. Again, now we want to do, because we want to do another one of these bloody chi-squares, right? Okay. So from this gets a little trickier, but you'll be doing an example of this. You want to do a chi-square test to determine whether this locus is in Hardy-Weinberg equilibrium. So one of the key things we got to come up, here are the observed numbers. We got those, right? So there were 400, 200, and 400. What we have to come up with are the expected numbers from the Hardy-Weinberg equilibrium model. And so it's fairly simple. The expected frequency is based on, you take 0.5, in this case you've got to take what that small p value is. You take 0.5, square it. Then you take 2 times 0.5 and 0.5, the values of the P and the Q, and then you take the Q squared to get kind of what we call you got to get the expected you got to turn those frequencies into numbers, right? And that's what we're trying to do here. So the first step is you got to come up with the kind of the expected frequencies. Okay. So what this says under Hardy-Weinberg, if these are the frequencies of the alleles in the population, the expected frequencies of the genotypes, you expect 25% to be this, 50% to be heterozygous, and 25% to be the other homozygote. Okay. How do you turn those into numbers? Yeah, so you just take each one of those expected frequencies and multiply by the total number of individuals in that population, so it's back to 1,000. So you always kind of got to go back to the total population size to get this. So here's what we've got. Under Hardy-Weinberg, we expect 250 to be this homozygote, 500 to be heterozygous, and 250 to be the AVI homozygote. Okay? Obviously, you can do all this, right? Now you do a chi-square test. That's a big number. Do those expected numbers look really similar to the observed numbers or pretty different? Looks like there's a big difference between the observed and expected here. So 250 versus 400, 500 versus 200, and 250 versus 400. There's a big discrepancy between the expected values under Hardy-Weinberg and what we actually see. And so again, the chi-square value, when you do all the observed minus expected squared divided by expected, sum them all up, you should, and you might want to do that as a check to make, you should get a value around 360. The degrees of freedom are 2 because you have 3 genotypes, right? And you reject from this, there's a really low probability that the observed and expected numbers are similar to one another, close enough by chance. So you really reject that hypothesis that this gene is in Hardy-Weinberg. Okay? Yeah. 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 That's from 2PQ, remember? Remember when you go back to the, yep, yep. So when you go back to that, remember that magical formula for the equilibrium frequencies of the genotypes? This thing right here, okay? So that is the 2 times the frequency of one allele times the frequency of the other allele, gives you the expected frequency of the heterozygote, okay? So that's 2, p is 0.5, and q is 0.5. So that's p squared over here, 2pq and q squared. Does that make sense? Okay. That's just kind of, so as we do this, if you did an example like this would be an example where this gene is in fact not in Hardy-Weinberg equilibrium. So I'm not going to go into what's actually happening here and what might be the possibility. Can you think about, can you construct an hypothesis to explain why this gene may not be in Hardy-Weinberg when you look at the observed and expected numbers? What looks weird? So you expect 500 heterozygotes, right, in Hardy-Weinberg. What are the observed numbers? 200, so they're a lot less. There are fewer homozygotes than you expect under Hardy-Weinberg. What about the homozygotes? That's just the opposite, right? You expect 250, and what you actually observe is a lot more. So what you see here, and I'm not going into the selection, I'll come back to this eventually, but anytime you reject a hypothesis, there must be some evolutionary process going on that's causing that thing to deviate from the equilibrium. And there might be some both selection and non-random mating that could cause this, and we'll come back to this. But what you see here is you find an excess of homozygotes, right, compared to expectations under Hardy-Weinberg, and you find a deficiency of observed heterozygotes compared to expected. Can you come up with can you construct any hypothesis to why you might expect, why that might be happening? What might be a real simple one? Non-random mating could be one. We won't get into that one. What about selection? What might selection be acting against in this population, do you think, against the heterozygotes? So one hypothesis we could construct here is maybe there's a deficiency of heterozygotes, right? Because there's something, they are under selection. Selection is acting against them. And maybe selection is favoring the homozygotes. So that might be one hypothesis. So as you go, this will be, so I don't think you do this in lab, but you'll do this on the homework. So do this, and it's not just plowing through all these calculations and doing this sort of stuff, but once you get the results, trying to interpret what might be. Do you reject the hypothesis of Hardy-Weinberg? If you do, then something must be going on. Okay, let's do an eye clicker. Excuse me. So here's the question. Which of the following is an assumption of the Hardy-Weinberg model? Hold on. Now remember, it's an assumption. So think back to the previous, the 5, right? Allele frequencies do not change over, this is a little tricky, but we'll see how you do. Allele frequencies do not change over time. Mating is random with respect to genotype, that is, there's random mating. Variation is introduced to the population by mutation only, and D, natural selection eliminates deleterious alleles. What we're saying, what Which of the following is an assumption of the Hardy-Weinberg? Okay. Everybody in? There's going to be another one, I think, later in the lecture. I think, maybe not, but okay. What's the answer? Yep, B is the answer. I knew. So A is. I knew that was going to be. So why is A wrong? Well, that's basically the outcome of Hardy-Weinberg. That's not an assumption, okay? So I knew that was going to be the tricky, the one that was probably likely. So that's basically, that says nothing about those 4 necessary conditions or 5 necessary conditions or assumptions. It's basically the outcome if all those assumptions are held true. So that's a little tricky. Okay. Okay, I'm going to stop this recording.